Happy Friday. Weekend once again. You just have to get through this class first. We are going to start talking today. We're going to finish up today with mRNA processing, post-transcriptional processing. We're going to talk about splicing, so I hope everyone is ready for that. And then I believe at the end of lecture today, we'll start getting into translation. We'll start looking at how to draw the structure of proteins. We'll pick that up on Monday. All right? The same set of announcements, just to make sure everybody gets them. Your first exam, you have until next Wednesday for a final point adjustment, so make sure that you come in and talk to me if you have questions. After that, we're done with exam 1 because it's time to move on to exam 2. The second exam, a week from today. There's 1 week left. On Wednesday, I sent out the study guide, and I posted the Mastering Biology practice questions. So those are all available for you. The lecture material is everything we cover up until through lecture on Wednesday. All right, and don't forget all exams are cumulative, so any questions from covering material from the last exam are still fair game. You can't forget any of the things that you've learned. So don't purge that information and replace it with the new information. You still need that. All right, to help you out, the TIs are going to have a review session again next Tuesday, 5:30 to 6:30 in this room. All right, so if you can make it to that, that is always a helpful thing to go to. And then because our exam is on a Friday, which just makes recitation awkward for that week, I'm going to cancel recitation for that week. So you have a week off of recitation. You have that extra time to either study for the exam or recover afterwards, depending on what day your recitation is. All right, are there any questions about the exam? Are there 1 or 2 at this point before we go on? Talk about post-transcriptional processing. All right, second exam is going to look an awful lot like the first. Some multiple choice, some drawing. You have to know your structures. You have to know your amino acids for this one. Make sure that you read all the questions carefully. You may have figured out after the first exam, the questions are kind of tricky. If you're reading quickly, you're going to miss something. Other questions? All right, say that again. Homework, yes. So thank you for reminding me. I am going to assign a short homework assignment for the weekend. It cannot be extra credit. It is to help you get into the material we're going to cover Monday and Wednesday of next week. So you start working with it now rather than next week, right before the exam. All right, so it's to help you get through some of that and to have looked at it before we cover it in class so you're not cramming for it last minute. So there will be a homework assignment. I will post that when I get back from class. It's going to be on translation. It'll be practice for you for going from nucleotides to amino acids, drawing your amino acid structures, right? Things that you're going to need to know for the exam, and it's good that you practice anyway. So I'm going to give you some practice questions in the form of a homework which you'll get credit for. Bonus, right? Yes. Homework keys for 5 and 6. Yes. Am I not posted 5 yet? Should have said something sooner. Sorry about that. I will post 5 that I owed you from last week, and I'll post 6 at the end of the day today. So you have those. Any other questions? All right, then we'll continue on have covered transcription. We've gone from DNA to RNA. We want to get to translation. We're going from mRNA to protein. We're on this step in the middle, but isn't really written into the central dogma. We are looking at how that RNA that's produced from transcription is processed and turned into mRNA through post-transcriptional processing. Just to refresh your memories, we talked about on Wednesday the difference between prokaryotic and eukaryotic transcription and whether or not there's processing. So the whole sequence, transcription to translation, and how that's different. All right, so you should be familiar with that. And we're talking about now how we get from this primary transcript, the thing that's transcribed from the DNA template, also called the free mRNA, how we get from here to an mRNA. So we have these regions that we need to cut out, we need to cap, and we need to put a poly A tail on that in order for it to be an mRNA. All right, so we're going to finish going through these processing steps today. We talked about, on Wednesday as well, capping. All right, you'll see lots of different representations of this. We talked about the important things, the methyl groups on the first two bases, the methylated guanosine, the 5' linkage with a triphosphate in between. All right, you have to have that when you're drawing your cap. All right, are there any questions on the caps before we move on to splicing? Okay. All right, so splicing. Excuse me. We talked a little bit about introns and exons. We're going to talk a lot about them today. We're removing those introns from that primary transcript. We're getting rid of those intervening sequences that aren't involved in translation. So we're cutting those out and we're stitching the exons, the pieces we want to keep, back together. So we're going from that primary transcript, cutting out those introns or those black regions, ending up with an mRNA that has an open reading frame contained within it. All right, so our first clicker question for today. Where does splicing occur? We talked about this on Wednesday. Where does this particular step happen in the cell? Everyone get their answers in? Let's take a look. Heidi? All right, so the majority of you say the nucleus, a few of you say the cytoplasm. Remember, in eukaryotes, where this processing happens, right, all of these processing steps doesn't happen in prokaryotes. All of these processing steps happen in the nucleus before it gets taken out of the cytoplasm for translation. So splicing occurs in the nucleus. And the suite of proteins and RNAs that accomplish this splicing are called the spliceosome. There are over 200 proteins involved in this process, and a couple of RNAs. Our SN RNAs are involved in splicing. All right, and the way that it works, if we look at an intron, we have an intron-exon boundary on both the 5' and the 3' side. Our exons are in blue, our introns are in red. Notice that, so there's that boundary, that splice site, which the spliceosome is going to cut the primary transcript at. And they're going to cut it at a very specific recognition sequence. So on the 5' side, all right, so this A or C represents that there can be some variation at that site. The important nucleotides are the big ones. So at the 5' we have AG, GU on either side of the splice site, followed by some variable sites. That R is part of the ambiguity code. So that stands for either A or G. So we can use single letters to represent multiple nucleotide options. On the 3' splice site, we have AGG. AG on the intron side, G on the exon side. All right, and these are very specific. We're going to see how this mechanism works. You have to have those specific nucleotides present at that site to be spliced. Yeah, Jessica. So G-U or C-U means that at that position, there's one of two possible bases. So it can either be C-A-G-G or U-A-G-G. So those are less important, those variable sites, than the giant letters, the A-G-G-U and the A-G-G. So there can be some variability in nucleotide, what the nucleotide is on either side of those splice sites. The important thing is that AGGU on the 5' side and the AGG on the 3' side. Notice that the splice site on both 5' and 3' has a GG on either side of that. Yeah, Jeremy. The R stands for either A or G. So when you're dealing with nucleotide sequences, how many people know what the ambiguity code is? Has anyone done any sequencing yet? No? So when you're dealing with sequences, if there's a site that is variable, you can write it either as A or G, meaning that at that position, you can have one of those two bases. Right? Or there's an ambiguity code that's standard for nucleotide sequences, where there are single letters that represent combinations of bases. So an R equals A or G, a Y equals T or C. What are some of the other ones? M, I think, equals A or C. All right, so you can look this up online. So if you see a letter in a nucleotide sequence that isn't one of your A, G, C, T, it's probably one of these ambiguity codes, and it means that at that position, it's variable. And that variability is represented by the single letter code, which actually stands for some combination of other nucleotides. You see an N, so you've done sequencing in your lab now. Some of you may have seen an N in your sequencing results. That means any possible base. We don't know what goes there. All right, wild card. All right, good question. Okay, so we have these splice sites, we have a recognition sequence, we have a spliceosome, is a combination of cellular machinery involving both proteins and RNA that are going to do this splicing. It's going to happen first. It's going to cleave that RNA, that single-stranded RNA, remember, so we just have to cleave one sugar phosphate backbone on the 5' end. First, second, it gets cleaved on the 3' end. So now we have a free piece of RNA. And then those two ends of the exons are brought together and stitched back together so that we end up with the two exons forming one mRNA, the intron in red being cut out and then degraded, gotten rid of. All right, so that whole process, cutting out that intron, stitching back together the exons. Notice that on either side of that splicing boundary, that AG on the 5' site gets stitched back together with the G on the 3' site. All right, we'll think about that when we start talking about intron exons and how they're spliced out and how you look and decipher the nucleotide sequence to amino acids. So, if every primary transcript that comes off a eukaryotic gene is spliced, then what's the intron exon pattern? All right, think about that for a minute. Read all those options carefully. Talk to your neighbors. Everything is spliced. Think about the process that we just talked about and how it happens, and what that means for how introns and exons are arranged in any gene. All right, has everyone had a chance to figure that out? Talk to your neighbors? Everyone get their answers in? All right, so if every primary transcript in the eukaryotic cell that is transcribed from a gene is spliced, let's look at what you think the pattern must be. All right, good. I think you have this figured out. That is correct. There we go. All right, it means that the pattern has to be exon, intron, exon. Just to really drive that point home, to make sure everyone gets it, these should be quick questions then. All right, so if I give you a picture like this, you should be able to tell me how many introns there are, how many exons there are, and can it be spliced properly? Yes or no? Remember, exons are in blue, introns are in red. Is this spliced properly? Yes. Let's take a look.
Oh. Yeah. Really?
All right. So the question up front is, if I ask you to draw this on the test, and you need to draw that AGG. If I want you to draw that recognition sequence, I'll be specific about that. Okay? All right, so can it be spliced properly? Yes? Darn it. All right. Can this be spliced properly? Just say yes or no. Why? Because why? Every intron has an exon on either side. Can this be spliced properly? Yes. Can this be spliced properly? No. Why? The intron doesn't have a 5' exon. Can this be spliced properly? No. Why? There's no 3' exon. Well, that's a good way to remember it. So, to help you remember that intron is the one that's spliced is because it wants to be on the inside of the exons. Or on the exterior. Thank you. Yes, there you go. Perfect. Good way to remember it. All right, so for splicing, the pattern always has to be exon, intron, exon. There has to be a 5' boundary and a 3' boundary in order to be spliced properly. All right, so does it matter how many introns there are? Is there a limit to how many introns a primary transcript can have from a gene? Get their answer in. Let's take a look. All right, so the majority of you thought there wasn't any limit. That is correct. Let's look at a few examples of that. So this, if there's no limit, what's the rule for the relationship of introns to exons? There's always one more exon. All right, so here are some examples of genes and the animals that they came from. So we have one intron, 2 exons, sort of the simplest case we can think of. All the way up to this Titin gene in the human has 233 introns that have to be spliced out. All right, which means there are 234 exons. So if I ask you to draw a primary transcript with a certain number of introns, make sure that there's one more exon in there. All right, so here's an example of that. Just to give you a picture, here's one with just a few introns, exons. Here's a gene with a bunch. So small, a few thousand nucleotides. Here's a gene that's 200,000 nucleotides that gets transcribed and then spliced out. All right, so huge variations in the way genes are put together for that primary transcript. All right. So we know that the primary transcript gets spliced. How does that happen? So we're going to look at the very specific mechanism, right? And it happens through a series, 2 to be exact, Trans-esterification reactions. All right, so here's the part where you have to pay attention. We're going to look at exactly how the spliceosome splices that intron out and the reactions that break that RNA and put it back together. All right, so here's where we're starting. Here are our recognition sequences on either side of the intron as our splice junctions. This is where we're ending up. There's one more important piece that we have to pay attention to, and that's there's an A, right, an adenine inside the intron that's going to play a crucial role in this process. All right, so we now have the splice junctions and an internal nucleotide to pay attention to. All right, our spliceosome composed of proteins and these SNRNPs, or SNRPs, small nuclear ribonucleic proteins. They're RNA protein complexes that are part of the spliceosome. All right, so they are recognizing these splice junctions and this interior adenine. And they're bringing the adenine, so they're creating a loop out of that intron and bringing the 5' splice site close to that adenine, to that base. All right, we're going to call this a lariat formation. We're going to make a loop. So if you want to draw it in our notation, here's our exon, intron, exon structure, 5', 3'. Flip it 90 degrees, exon, intron, exon, 5', 3'. There's our splice site with a guanosine on either side, our adenosine in the middle. All right, we're going to draw that adenosine in the middle of the RNA, and you want to draw that ribose. All right, remember we're dealing with RNA and what that means for the sugar. And then what the spliceosome is doing is it's bringing that adenosine close to that 5' splice site and making a loop. So we're going to take this drawing, turn it into that drawing. All right, so it's creating this loop by bringing this adenosine close to these guanosines. All right, keep track, 5', 3' still. All right, and the other schematic that we looked at is the spliceosome that's bringing it together, and then we're going to get cleavage at that 5' splice site. All right, the way that that happens is if you've drawn that ribose, so everyone draw a ribose hanging off of an adenine. So draw yourself an adenosine in the middle of that loop structure. Put the 2' hydroxyl on it. Remember, it's an RNA. And then we're going to make a 2', 5' phosphoester linkage to a guanosine. All right, what is the normal phosphoester linkage in our nucleic acid backbone? 5' to 3'. All right, so this is a new linkage. We're going 2 to 5 rather than 3 to 5. All right, let's take a look at what that's going to look like. So here's our lariat structure again. 5', 3', exon 1. Here's the G and the G at our boundary, our 5' splice site. Here's our 3' splice site. Here's our internal adenosine. Here's our 2' hydroxyl. Here's our normal 5' to 3' sugar phosphate backbone. All right, so the spliceosome brings this adenosine close to the splice site GG, and it's going to flip from a 3' 3,5 between the G's to a 2,5 between the A and the G. All right, so we're going from this, right, to this. Everybody see that? I'm going to flip back. All right, here it is before the transesterification reaction. Everything's normal, just looped around and close to each other. We're going to form this bond now and break this one. So we're flipping our 3' to 5' sugar phosphate linkage between the two G's to a 2' to 5' linkage between the internal A and the G in the intron at that splice site. That is our transesterification reaction. We're just flipping where that phosphate is bound to. All right, everybody with me so far? Yeah.
Is there a particular reason why there's no hydroxyl group on that on the previous side?
Yeah. Mostly just because I forgot it. Good point. Thank you for catching me on that. I'll correct that. There should be, right? So this is RNA, so there should be an OH here. I didn't draw it because I was mostly concerned with what was happening up there. But you're right, there should be one there. All right, any other questions before we proceed? So at this point, we've cleaved that 5' splice site. And we formed a loop. So we have our 3' end that wraps around and then binds back into the middle. That's our lariat structure. All right, so where is your free hydroxyl now? In the guanosines. We want to go now and splice that 3' site. All right, so we're going to have to reorganize our drawing a little bit. First, let me show you in the schematic. So we formed our lariat structure here. Our spliceosome now brings that 3' in. All right, so this is where it can get a little confusing. It's the 3' end of our 5' exon. All right, 3' end close to the 5' end of our 3' exon, just to be extra confusing. All right, so it's going to bring this end close to this end. We have a free hydroxyl now on the guanosine that we just split. All right, so here's where we're at in our line drawing. We have that lariat structure, and we have our free 3' guanosine on the first exon. If we redraw it so we can now see the guanosines at the 3' exon boundary. Right, so all we've done is flip this 90 degrees and drawn the guanosine here. And our second exon. So what we want to do is put our exons together. How are we going to do that? What linkage? 3 to 5, 3' of what? The 5' exon guanosine to the 5' phosphate of our 3' exon. Everyone see that? Taking that and putting it over there. And then so we're going to flip again, we're going to flip that phosphate bond, that phosphoester bond, from the backbone at that splice site to the backbone on the exon, first exon. So we have our second trans-esterification reaction. So it takes two of them, one at each splice site. The first one forms that lariat structure, so we're flipping the phosphate on the intron side to an internal nucleotide. The second one both splices that lariat structure off of the second exon and puts it together with the first exon. So it takes two steps. Splice the 5' end, and then put that in together with the next exon, which splices the intron out. And then that lariat structure can go on to be degraded. All right, so this happens to cut out every single one of those 233 introns that were in the Titin gene. Or the one intron that was in the, was it an actin gene? I forget what gene it was now. All right, so you should be able to draw the branch points involved in these two separate reactions. All right, and that branch point is going to involve the standard 5' to 3'. All right, so we've got this adenosine, got a 5' to 3' linkage, 5' to 3' linkage, as well as that novel 5' to 2' linkage, what makes our loop. So you should be able to draw this branch point. All right, so here's the whole process. Spliceosome brings the 5' to that internal adenosine. We get our first transesterification, and then that free end of that side is brought to this splice site, and we get our second transesterification, which joins these two exons and cleaves off our lariat intron. Right, and you should be able to draw this branch point here. This is our branch point, that adenosine. 5', 3', 5', 3', 5', 2'. Okay, that is splicing. Everyone got that? Just like that? All right. Two transesterification reactions. Be able to draw that branch point. Be familiar with that novel 2' to 5' linkage.
What exactly are the branch points? Like, what specific point is this?
So, but when I say branch point, I mean this adenosine, which is both part of the normal backbone of that intron, as well as where we get that transesterification and bringing in that novel 2 to 5 linkage. So normally you just have, 3, 5, 3, 5, 3, 5, but here you have a branch point. You've got something coming off the other direction. So where the lasso. So exactly, where that lasso comes together. That's the branch point. You need to be familiar with all of those steps. You should be able to draw them. But this is the really confusing, tricky bit, I think, in all of that. So you need to be familiar with this branch point, right, in that lariat structure. So you need to be familiar with the steps, and if you understand this, then you understand what's happening in all those other steps. So this is a representation of the same thing. All right, so this is the branch point. So here's our normal backbone, and here's the link of the 5' into that loop. Same thing. Right? But all I'm showing here is that 2', 5' linkage. Well, unless I give you these other bases, right? Then if I ask you to draw that branch point, I'll be specific. Like if I ask you to draw the 2', 5' linkage in the lariat, you can draw it this way. That's the 2', 5' linkage. Okay? Question?
Is it okay to just use the letter A and B and draw out? Or are you going to specify?
I'll specify that on the exam, so I want you to draw bases. Any other questions? You should certainly by now know how to draw the bases if I ask you to. Any other questions about splicing? Here is one link to a YouTube video that goes through that process. If you'd like to watch somebody else explain it to help really cement that in your brain, that will be available on Facebook as well as in your lecture slides when I post them. So what are we still missing? Poly A tail, right? So we've spent most of today on splicing. Let's just go quickly through the poly A tail, and then we're all the way through processing, and we can start on translation on Monday. All right, so this is pretty much it. We have our 5' cap. We splice out our introns. Then we have our 3' end of our primary transcript. There's a recognition sequence. This ARIT, AAU, AAAA, where that primary transcript is cleaved. So the 3' end of the primary transcript is cut off. And then a poly A polymerase, an enzyme that just puts A's onto the end of the strand, comes in and gives us a poly A tail. Similar to the 5' cap, this poly A tail protects the mRNA from degradation once it's moved out into the cytoplasm. The longer the tail, the more stable the mRNA. Right? The more likely it is to persist and not be degraded. That poly A tail is also involved in recognition for export to the cytoplasm and in recognition by the ribosomes as an mRNA for translation. All right, so the poly A tail is important. It's a quick and easy process. Cut off the end and add some A's with an enzyme. But it's very important for that mRNA to have. All right, so I'm going to leave you with this question today. We'll pick up here on Monday. Everybody have a good weekend. I will post a homework to help you practice that lariat structure, to help you practice translation, and what we're going to get into next week before the exam. If you have any questions, come and talk to me at office hours or talk to your guys. And we'll see you on Monday.